like to thank meeting organizers for giving me this opportunity to present my research result. Yes. Can you hear me? Okay. I feel I come to talk to a broad, the audience with a broad background. So I try to slow, and I appreciate you ask me any question, even in the middle of talk, so that will help me to deliver my research result more informatively. So this is a very general title, and I actually will give slides according to this sequence. So I give the introduction. I want to show you the Lesquerella seed development, all the processes, and how is hydroxy fatty acid synthesized. And then I talk about seed transcriptome. It's a big subject, and we have published. So I try to cut this little bit shorter in order to give me more recent results. How about the gene expression and transgenic work, and which is very exciting at this moment. So conventional hydroxy fatty acid is come from castor oil. So this structure we call the TAG, triacylglycerols. It has a glycerol backbone and acylated with 3 chains of fatty acid. Most vegetable oils has this kind of chain, but without this hydroxy residue. So it's called the oleic acid, but with this hydroxy residue, it's called the ricinoleic. Because of this residue that make castor oil very high viscosity, and it has special physical and chemical properties that make lots of product different from equivalent product from petroleum raw material. For example, the lubricant made from castor oil has high lifetime quality, also can withstand very low temperature. So it's been unique lubricant only used in the aviation or submarine engines. And there are also applications of these fatty acid can turn to polymers. Basically, all the petroleum product can be made from castor oil fatty acid. So we can see all 3 positions, 1, 2, 3 of glycerol backbone are acylated by this particular fatty acid and at a high concentration, like 90%. So this is a valuable source of castor. However, we do not grow castor in US. We imported all the leads. So the major problem is the toxin ricin. And second is triacylalbumin. These are also very potent allergens. So because of this hazardous material, there's a lot of resistance actually from political people level, at the ministry level, also from farmers. They don't want castor grower be in US because lots of contamination of the bean goes to other product like a grain or the corn or the soybean. So that's a reason. So one of my projects is try to develop a safe domestic source that can replace castor oil import. That's why I work on Lesquerella. So the Lesquerella basically is a parasitic species and looks like an oilseed ripper in height, but the seeds are very small. It has about 60% hydroxy fatty acid. And this fatty acid mainly is the 20 carbon chain hydroxy fatty. It's a longer, just one more step elongate into this 20 carbon. And second issue is the middle chain. It doesn't have a fatty acid, has any hydroxy fatty acid. It just has a oleic, linoleic, and linoleic acid. So these all unsaturated fatty acids are ester in the second. So there must be a difference in the mechanism of the oil synthesis pathways between castor and the Lesquerella. And one of my goals is try to investigate the difference of this mechanism and try to manipulate the Lesquerella that be able to produce castor oil like TAG. So this is seed development. So briefly, we just can see it takes 49 days after pollination. So at the early stage, let me show the stage. So basically, the content are mainly water. So oil start to synthesize at age 21 and start to accumulate rapidly and go to high, relative high at 35. So 21 to 35 is a mid stage of seed development. And you can see the fresh weight gain rapidly at the early stage, but mostly water, 21, 40. So remember, these are very early stage. And we want to see the hydroxy fatty acid accumulation pattern. So these are common fatty acids we see in most vegetable oil. So they exist in Lesquerella, but during the seed development, you can see the content changes. But let's focus on first hydroxy fatty acid, ricinoleic acid. It does exist in Lesquerella, but at a very early stage, it starts to accumulate 17%, which is quite a lot. And right after a week, these hydroxy fatty acids dropped. And the second, the longer hydroxy fatty start to accumulate. And these lesquerolic acids continue to increase to the end, become a major fatty acid. That means the fatty acid, once this substrate, ricinoleic, once substrate is accumulated and elongates, is strongly activated to elongate these hydroxy fatty into a lesquerolic acid. And the elongase is so strong, it just keep push all the produced ricinoleic into lesquerolic acid. That's the difference. There's strong elongase in lesquerolic seeds. And there is a desaturase. So whatever the desaturase, it's not strong, it's weak. So desaturase can desaturate ricinoleic, also can desaturate this 20 carbon chain oleic acid. So desaturase of ricinoleic show little called the densopolics. Detected a minor desaturation of these lesquerolic produce oleic acid, and it also accumulate to certain level. You see the sequential, it's just to tell us that it's the pathway going and the enzyme activity follows consistently with the synthesis process. So see the transcriptome, something may be cut. It's a collaboration with Dr. King from RDA at the Korea. So why we do see transcriptome? So we can produce genome-wide sequence data for expressed genes, and we can provide global view of the activity of the biology process, and give relative expression level for different genes. I will take example of that. Allows rapid identification of a whole set of genes involved in the specific metabolic pathways. For me, particularly interested in as pathways for fatty acid and TAG biosynthesis. So last not the least is help on genome annotation. In future with genome sequence, these ESDs can help to tell where the genes are located. Yeah, so these are results come from this paper. Kind of summary. So we have total 59,914 transcripts identified from original 651 megabase raw data. And these transcripts represent 20,995 unique genes that include nearly all known seed expressed genes. So which is a very good seed transcriptome raw data set. And 74% of these unique genes matched with known genes. So we can classify into different groups and know their potential function in Lesquerella. I think among these known genes, we show 90% of them with the highest homology with the rhabdomycin gene. So that means they are very closely related between these two species, In all the molecular work, genetic work done in the lab analysis, we can borrow the result and we can even interpret the result to Lesquerella. So we can do translational biology in Lesquerella. So this is an excellent opportunity for AIS scientists to work on the crop that's so closely related to the Rhabdopsis. So I do not want to show all the details. I highlight blue is the one I'm going to show result in the table. Basically, there are 16 categories of lipid metabolism classified by Rhabdopsis community. And fatty acid synthesis, total rhabdopsin has a 43, but expressed seeds has 28. In Lesquerella, we found 29. They are very close, even the number of expressed genes. Second group, elongation desaturates exports from plastic. That's mostly happened in the ER or the cytosol. And Rhabdopsis expressed the seeds found 13, we found 22 unique. And the third is oil synthesis. Try also grocery sample that happened in the ER, and we can see Rhabdopsis, they put on 69, we found 59. So this data give us a very good confidence to explore our new experiment. This is a picture from textbook. So introduction for oil synthesis is plastic is the location for fatty acid synthesis in elongate. So palmolic acid and oleic acid, they just activate into CoA in the form of the CoA. And this fatty acid CoA, sometimes also called acyl-CoA, are integrated into membrane of ER. And in there, they try to assemble into TAG using glycerol backbone. So that's the area I will focus of my talk in the next few slides. So traditionally, there's one pathway, especially many years ago, we called the Kennedy pathway or conventional pathway. So glycerol phosphate, the backbone has been, is a precursor. And those acyl-CoA pole, those fatty acid CoA is a mix of fatty acid. We just acylated into number 1 position by GPAD, and then go to second position by LPAD, and then third position by DGAD. So these are the key for these. So after 1, 2, 3 steps, and all the glycerol bond is acylated by fatty acid. So there are specific enzyme work on the specific position from this slide. So LPAD is responsible for synthesize fatty acid at the SYN2 of the position. Remember the difference between castor oil and Lesquerella oil. LPAD is the one I feel most important for the engineering reason. And this is a castor oil, this is a Lesquerella, just to review. That's why I want to put these slides there. Now, how is hydroxy fatty acid synthesized? It's on the PC. So once the PC has an oleic acid, the oleic acid can be desaturated into linoleic acid by FAD2 and furtherly desaturated by FAD3 to linoleic acid. So there is a called hydroxylase. Found first in the castor, and Lesquerella, they also have a similar hydroxylase. They make hydroxylation on the oleic at the SYN2 position. So I will show you a slide how these hydroxy fatty acid being released and go back to the acyl pole and go to the Kennedy pathway and become a dominant in the oil in the TAG. So this is what we call the acyl editing. So they can be a kind of a modifier for the de novo Kennedy pathway. So fatty acid is synthesized in the plastic through the mechanism we already showed. And it activated into a CoA. And this CoA actually is quickly imported into ER by LCAP. So this LP cap is just rapidly create lots of called modified PC, and it has modified hydroxy fatty acid at the syn2 position of this PC, including hydrosylation and desaturation. This modified hydrosylation was exported back to the acyl-CoA pool, which is local in the cytosol. So this pool become more and more complicated during the seed development with all the enzyme activated. And an elongate called the ACS18 will elongate ricinoleic CoA into Lesquerella as CoA. So that's how the elongation, how this hydroxy fatty acid being made in the cytosol. And if you remember the Kennedy pathway, I put the Kennedy pathway in here, a little bit of curving down. So first the enzyme GPAD in the Lesquerella seeds, they are dominant with Lesquerella acid. They just put a GPAD, GPAD will use Lesquerella acid as a acyl donor. And however, LPAD2 seems excluding the Lesquerella acid. So LPAD doesn't put any hydroxy fatty acid in there, but DGAT also not discriminating and put the hydroxy Lesquerella acid. So I'm not sure GPAD and DGAT is selectively doing that. At least they don't discriminate. And LPAD in Lesquerella looks to me try to avoid, because Lesquerella is a dominant fatty acid. In the acyl-CoA pool, these will become more and more at the later stage of development. And no matter what, they just don't pick that hydroxy fatty acid. So that's very interesting. Since we are omics topics, I want to show how is omic genome data and gene expression analysis can help us on engineering side, which is applied side of the science. So I want to show example highlighted in red and blue. So if we talk about DGAT, we found 4 DGATs from the seed transcriptome. How can we know which one is a major enzyme to work on the Lesquerella acid, the hydroxy fatty acid? And LPCA2, that's one we see. It's also a key enzyme because they actively generate these hydroxy fatty acids or modified hydroxy fatty in the pool. So they have 2 enzymes. So we want to know which one we want to be future target. So for example, if we do that, we do DGAT and we see the expression pattern, we can easily say DGAT2 is not seed-specific. They express high in the leaf and stem also in the early stage of seed development. And this one, DGAT3, we know it's a cytosol specific. It's not membrane bound, not at the ER. So no much surprise. We see lots of leaf, stem. But these 2 enzymes draw very high attention, especially DGAT1-2. It's particularly increased with the seed development and relatively low in the flower buds. Looks like flower buds has lots of oil. So lots of lipid gene also expressed high in the flower. So, but this one is more like consistent with oilseed synthesis. And this one looks like a leaf stem is same high as the early stage, but only high at the very late stage. And looks, and to me, may be involved in the particular seed coat development wax synthesis. Very different process. So this kind of analysis will help me try at least candidate 1 or 2 to do our engineering work in the future. And people interested in the Camelina already asked me for the gene of this particular DGAT because they think they want to use the DGAT to produce hydroxy fatty in the Camelina. And Camelina is close to Lesquerella, or rather close to custom. so they are more distance. So this second example, we can see this one has high expression in the flower bud, but in the seeds they start to declining. They are earlier, but the decline early is more like a vegetative tissue, lots of water in the seed, not much embryo in storage compound. But this one is something we think that's the right candidate. In the future, we want to enhance the expression. We want to look at these. So biochemistry is very important, but molecular at this point is more specific for us to identify the key regulator. So now this part, I originally did not put the abstract when I submit, but it's my recent result, very exciting. Some of them are still in a preliminary. So why we do biotechnology for Lesquerella? We try to increase the seed oil content and yield. That so far is lower compared to castor. And we want to increase hydroxy fatty acid level in the seed oil. As you can see, it's missing 1/3 compared to castor oil. So produce uniform oil. So it is an industrial crop. So we are allowed to manipulate hard to do all kind of things we want. For example, all kind of different hydrophilic, because we know the pathway, we can block and we can overexpress. We can try to make all the hydrophilics in the way we want. And it can produce other hydroxy fatties, introduce genes from other specific species, and make Lesquerella crop is more useful. So this is the first successful engineering project I have done. So first of all, I identified castor LPAT2. That was, at that time, it was biomath informatically analysis said this is a gene work in castor through gene expression, through sequence alignment, but never has been put in the vivo to test its real function. So this is about 3-4 years ago. I found this gene, then I put it in a seed specific promoter, A promoter, and this is a transformation vector. I transformed it into Lesquerella, and this is a wild type. I'm talking now is the hydroxy fatty acid racemolic expression by expressing castor LPAT. You remember castor has all the 18 chain length. So wild type has about 1.5%, but you see all the expressions, especially the red one, can increase more than 3- and 4-fold. So among 16 lines, except the line 6, I'm not able to continue because it died out in the middle. All the lines I see increased, and this is F1 seeds. It's a mixed population. So this is very exciting. The castor gene does something in Lesquerella. Now, I want to see if this increased hydroxy fat is at the SYNC2 position. Yes, it is. So the racemolic is extremely enhanced in Lesquerella compared to wild type, and this is more than 6-fold. And other hydrophones also increased. It's basically depending on the level. So what I want to say here seems castor LPAT is very selective to racemolic acid, and is also capable to isolate these hydroxy fatty in other crop, other species, for example, Lesquerella. So as a consequence, you have more in the SYNC2, the tri-isoglycerol, remember 3 chains in a glycerol backbone, all has a 3 position with isolated with a hydroxy fatty acid. This hydroxy fatty acid, TAG, content increase. So imagine in a wild type, only 1 in 3 has hydroxy fatty. In the transgenic, there are TAGs has 3 positions all isolated as a hydroxy fatty. So this is already a breakthrough. Say this gene worked, but how much, how good, and whether other pathways block it doing a better and further job. So we will see in next few slides how can we to improve in the future. This is a busy slide, but you already see, and this is the hydroxy fatty acid circulation in and out and going to the pool and aspirate to that. Okay, so this now after that, if I say, I show the example. If I put another gene, and this hydroxy fatty acid ricinoleic will be acylated by castor gene, and this is transgenic Lesquerella TAG pool. It still has lots of unsaturated fatty acid, but it has some hydroxy fatty acid in there. Now, which gene really make lots of these unsaturated fatty acid? I put the green because I think we can correlate from visual. Green is the enzyme PDCT, and that really push this unsaturated hydroxy into TAG through a pathway bypassing the kinetics pathway. So it do no longer follow the classical pathway we always think. It just, once the PC has a desaturated fatty acid, this PDH just transfer PC into PC DAG and go to TAG directly. There's no number 1, number 2, number 3 acylation process. Since PC is modified at the second position, desaturates, all these modified fatty acid at the same 2 position will go to the TAG without being removed, go back to cytosol pool. So this is a direct process. It's a short pass and quick pass. So this is an obstacle for even Rabidops people, Gamelina people, and me try to think how should we increase. So one example is if we put a castor PDGC in Rabidops work, it does show this PDGC will push the hydroxy ricinoleic fatty acid goes to PC. So in my case, I think, I'm not sure I showed this. Okay. So I show a couple of cases. I think I forgot to put a cross in here. I want to next step to put a castor PDCC, but block endogenous Lesquerella. So we just have foreign castor PDDC to work on hydroxy fatty. This has to be blocked. In second block place, you can block FAD3 at the early stage. So basically, I want to remove or reduce this unsaturated fatty acid at the very beginning. So make all the acyl-CoA pool relatively enriched with castor oil, like hydroxy fatty acid. And if we block elongates, so the whole pool will be remained like a castor oil, ricinoleic fatty acid. So that's a way to engineer Lesquerella producing castor oil. So this is, but to me, at this point, Lesquerella acid, 20 carbon chain, is a valuable fatty acid. And they are a chemical company. They particularly wanted this hydroxy fatty. It can create new raw material. So we can either produce castor oil like or produce Lesquerella oil, but with much enriched ricinoleic hydroxy fatty acid content in the seed oil to increase the overall content. So that's my future goal. I want to acknowledge people work with me, Kumi and Ava. They are the working student, and for a long time they are part-time student. They help me tissue culture, greenhouse work. And Roger is the one I collaborated with, construct the transformation factor. And Dr. King is the recent collaboration on seeds transcriptome, and John and Harry and Charles, they are the person I contacted with for doing fatty acid analysis, because my lab do not have that high capacity for hydroxy fatty acid analysis. Thank you for your attention. If you have questions, I will be happy to answer. Yeah.
Congratulations, excellent work. But let me see about how do you analyze the fatty acids? It's by GCMS, and you hydrolyze the triglyceride fatty acids. You have the fatty acids as an ester with triglyceride.
Yeah.
How do you hydrolyze to analyze the fatty acid?
Some of the results come from GC analysis. They will chop off all the fatty acid away from, they call the methylation process. They will, and go to GC. In some of the work, they have to use lipase and to remove chain 1 and chain 3 and only remain middle chain.
Yeah.
And they were able to see how much and what fatty acid in the middle chain.
Okay.
So they are actually quite a different method.
Yeah. But because I'm afraid, because if you hydrolyze, sometimes you can eliminate the hydroxyl group. Then when you quantify, you have less of hydroxyl fatty acid. Did you understand me? I'm a chemist, I'm not, I don't work
So
in
your
genetics.
last question is?
Because when you hydrolyze the triglyceride fatty acids, you use, for example, acid, then you can eliminate the some hydroxyl groups from the fatty acids. And when you quantify, maybe you had a hydroxyl fatty acid. But when you are analyzing, you lost the hydroxyl. You eliminate the hydroxyl group.
Yeah, that part probably I need to ask my collaborator for the detailed technique. But these people are all work on hydroxyl fatty acid in Camelina rhabdopsis. So they know what they are looking for. So yeah, I think the analytical part was probably
Thank you.
Yeah, I have a question. Grace, you were talking about the genome size of this Lesquerella. So it's approximately 60,000 genes, transcripts you have found. And out of the 60,000, you have 27,000 transcripts in the seed, specific, right? So, but my question is, how many genes or how many transcripts do you see which are involved in biosynthesis of fatty acids or triacylglycerol?
Yeah. There is a slide.
Yeah.
I may go back. I think this is the slide. How many genes? So this is how many genes expressed. Totally is this. Number 615.
Which are involved in biosynthesis of fatty acid in Lesquerella?
Oh, if it's a fatty acid pathway, 29.
29 only.
Yeah.
Okay.
29. In rhabdopsis, they found 28.
But these are the number of transcripts, right?
Yeah, these are unique gene transcripts
okay.
after a sample. After a sample.
Okay.
Yeah. Yeah.
Thank you.